All right. So my name's Jonah Jodlowski, and this summer I was also in Santa Barbara studying marine science. So what did I research? I researched sea urchins and algae and the relationship between them. So the two different species of sea urchins that I researched were Mesocentris fasciensis and Strongylocentris purpuratus. So this is a sea urchin right here. These are its spines. They actually really hurt when you're trying to pry them off walls of the holding tank. But on the other side of this is the sea urchin mouth. So actually what it does to eat the algae is it hooks it on to the spines on its back and then actually moves these spines and pulls the algae around to feed it into the mouth area. So when we later on did tensile testing, this was one of the reasons we did tensile testing, because it was easier for stretchier algae to be pulled around and fed to the sea urchin. So these are the 7 different types of algae that we worked with right here, or more, I don't know, I didn't count them all. And then fun fact, real quick, seaweed's not actually a weed or a plant, it's actually algae. All right, so the Santa Barbara Coastal Long-Term Ecological Research was the plan that I worked with. And so these are marine protected areas off the coast of Santa Barbara and the Channel Islands. And in these marine protected areas, there is no fishing allowed or capturing of sea creatures. And so the purpose of the Santa Barbara Coastal Long-Term Ecological Research is to study the effect of algal functional traits on the palatability by red and purple sea urchins. And this is because red sea urchins are actually harvested for food throughout California, while purple sea urchins are not. Red sea urchins are the bigger ones, and there is a clear difference in color. This is the research team that I worked with right here. Katrina was my mentor, and she is getting her PhD in marine biology at UC Santa Barbara. So the experiment. So step 1 of the experiment was preparing sea urchin holding structures. These were the cages that sorry, it's a bad photo, but these are the cages that we kept the sea urchins in during the entire feeding trial. And so we drilled holes in them so that seawater could actively run through them and nourish the sea urchins. Step 2 was measuring diameter and weighing the urchins. So we did this actually before and after the feeding experiment so that we could see which algae was most nourishing to each sea urchin. So step 3 was weighing and dehydrating the algae. So we would weigh 40 grams of each type of algae to put in each holding tank so that every sea urchin got the same amount. And we would also cut out different types of algae, or different shapes of algae to put into our dehydrator that would later be grinded to be put into a vial for CHN testing. And we want to test the elemental frequency in each different type of algae so that we could see which element they preferred. Step 5 was urchin feeding. So we would put them in their cages with the 40 grams of algae, and we would weigh them down in these holding tanks that had free-flowing seawater through them. We had to put weights on top of them because a lot of them would pry they're not that, the sea urchins are very large compared to the tanks, so they would pry the top off and escape through the tubes, like in Nemo. So step 6 was cleaning. About midway through the experiment, the water would get really, really muddy with sea urchin waste. So I would take our little siphon right here, and I would suck it up. About halfway through or actually about a week left in my internship, I realized that you don't actually have to suck it. You can actually just put it in the water and cover it with your hand for suction. But by that time it was too late. I don't know why anyone didn't tell me that. But so step 7 was variable because we either had a successful experiment or a failed experiment. These cages were very small, and most sea urchins did not thrive in them. So a lot of time, for the first 3 experiments, enough sea urchins died that we could not use our results. But the last 2 were successes. But after the completion of the experiment, we would sift their waste and put those in vials to send to the analytical lab for CHN testing. And then this was the CHN testing. These are the vials that we put them in. And we did not actually do this ourselves. We sent them to another lab to do. So I was not able to see the results when I was there, but she sent me them afterwards. Step 9 was post-feeding analysis. So this is what we did after we measured the post-feeding weight and or sea urchin diameter. And so our results, as you can see here, Macrocystis porifera, which is also giant kelp, the main food source for red and purple sea urchins, and Chondrochanthus corymbiferus were the 2 algae species that that nourished the urchins the most, that were most palatable to the urchins. So what I learned that these are passionate people that you work with. But the first day I was there, we were talking right when I met Katrina and John, and they were talking about Viking funerals and how they wanted to have them, and they were talking about how her preferred way to die was getting eaten by a shark so that her remains could go to nourish the marine ecosystem. I was like, that's interesting. I also learned that there are always mishaps. Science is science. It doesn't always work. Our first 3 experiments failed, and I would be collecting our dead urchins and having to throw them in the dumpster. But that's going to happen. And then even though it's a world-class institution, doesn't mean everything comes easy. We're still scrubbing tanks every day. Marine biology isn't for everyone. I loved it. Working in a lab 8 hours a day does get a little repetitive, though, because you're doing the same thing over and over. But if you are over 18, you do get to go on the dives, the surveys of the ocean, which is probably a lot more pleasant. And then the last thing I learned was that we have top-notch people watching over our oceans. These are people that attended world-class undergraduate universities. And so, yeah, outside the lab, these are a couple pictures I have. I'm running out of time, so I won't explain them all, but here's a soccer game that me and Justine went to. I still haven't told my host family that I snuck out for the soccer game, but if they see this, they'll know. And then these are all the people I would like to thank. Big thanks to Sarah and the whole Pinhead board of directors.
Yay, you did great.
Thanks.
Yay, thank you so much.